Welcome, everybody. We are ready to start the San Antonio Breast Conference. A few housekeeping. Your app will update immediately at 8 o'clock. We are going to have a hybrid session, so as we stumble through this a little bit, please bear with us. As Dr. Kakamani said, there were some cancellations. So it was a great pleasure to introduce the very first speaker, Dr. Bianchini, who will discuss single-cell spatial analysis by imaging, mass cytometry, and immunotherapy response in triple-negative breast cancer in the NEO-TRIP anti-PD-L1 trial. Dr. Bianchini.
I would like to thank the scientific committee for selecting our work for presentation. This is my disclosure. In NEO-TRIP trial, patients with triple-negative breast cancer were randomized to 8 cycles of nab-paclitaxel carboplatin with or without atezolizumab. In the per-protocol population, including only patients available for PCR, a modest 4.6% numerical increase of PCR was in favor of atezolizumab arm. We successfully performed imaging mass cytometry on 86.6% of pretreatment formally fixed paraffin-embedded biopsies. RNA sequencing was also available for 97.5% of these patients. 43 proteins spanning cancer cell and the tumor microenvironment were assessed using IMC on a biopsy section. A second biopsy section was stained with HNA. Samples were labeled with antibody conjugates to histotypically poor rare hair metal reporters and profile at 1 micro resolution by IMC using fluidine Hyperion. For each sample, we have generated high-dimensional images for 3 macroscopic fields that encompass the tumor and the tumor-stroma interface. By using various computational approaches, we first performed the cell segmentation step on all the images, allowing to identify individual cells. Then we assign each cell to the epithelial of tumor microenvironment compartment. By clustering, we identify 20 robust cell phenotypes within the TME, and 17 cell phenotypes were identified within epithelial cell. This is a graphical representation of the resulting cell phenotype annotation of this specific sample. In our study, we aim to evaluate the association of protein expression of each biomarker independently in the epithelial and TME compartment, cell phenotypes, density, spatial metric of cell-cell interaction, and we will correlate with PD-L1 status, stroma tiers, TNBC types, and PCR. This graph describes the association between proportion of cell phenotype by PD-L1 status. The size of each circle is inversely proportional to the standard error, and the black outlines correspond to P less than 0.05. Several cell phenotypes are differentially frequent in PD-L1 positive and negative tumors. Large androgen receptor positive cell and stroma cell, such as fibroblast, myofibroblast, are strongly overrepresented in PD-L1 negative tumors, whereas cells of the adaptive immune system are more frequently found in PD-L1 positive tumors. As expected, epithelial and TME cells expressing PD-L1 are largely predominant in PD-L1 positive tumors. The association of cell phenotypes with stroma tiers shows an even broader degree of heterogeneity. Considering the epithelial compartment, each TNBC type is heterogeneously rich by specific cell phenotypes. For instance, basal-like 1 has a higher proportion of cells expressing PD-Liga1 at MHC1 and 2. Large subtype is very highly enriched, as expected, in androgen receptor positive, but also in GATA3 positive cells. Mesenchymal subtype has more cells with IMT and neuroendocrine phenotype. Considering the TME compartment, both basal-like 1 and mesenchymal stem-like are enriched in CD8 and CD4, but only basal-like 1 is enriched in NK, plasma cells, and PD-Liga1 positive antigen-presenting cells. Instead, both mesenchymal and LAR types are enriched in stroma cells. Then we evaluate the association between protein expression assessed by proportion of positive cells in each compartment and PCR. The analysis was performed by treatment arm, and resulting odds ratio for atezolizumab arm are described in red, and for chemotherapy arm in gray. Black outlines indicating a p-value less than 0.05. Overall, more biomarkers were predictive of PCR in atezolizumab compared to chemotherapy arm. For instance, high Ki67 assessed in TME cells was strongly associated with high likelihood of PCR only in atezolizumab arm, resulting in a p0.06 for the test of interaction. The effect was also present, but less evident for Ki67 assessed in the epithelial cells. Conversely, HLA-DR was informative only when assessed in the epithelial compartment, but not in TME. Then we evaluate the association between cell phenotype density and PCR independently in the two compartments and by treatment arm. Again, high density of several cell phenotypes were significantly associated with higher likelihood of PCR in atezolizumab, but not in chemotherapy arm. In particular, antigen-presenting cell positive for PD-Liga1 and HIDO, and CD56 positive neuroendocrine epithelial cells have a significant test of interaction. Finally, we have also evaluated the heterotypic spatial connectivity between epithelial cell and each specific TME cell phenotype. In this assessment, only tight contact between epithelial and TME cells were considered as interaction, as illustrated by red cells in the figure. Here we illustrated 3 examples for low, medium, and high degree of interactions. By assessing the association between the degree of interaction of each TME cell phenotypes as continuous variable and PCR, we found a clear different pattern of association by treatment arm. Notably, for any marker, the degree of interaction is significantly associated with PCR in the chemotherapy arm, whereas 9 biomarkers were significantly associated with PCR in atezolizumab arm. The test of interaction results significant for 6 biomarkers. To better interpret these findings, these figures describe the association with PCR of the degree of interaction for CD8 exhausted T cell expressing PD-1 above, and for CD8 expressing granzyme B below. The association with PCR are illustrated by treatment arm, confirming a predictive value only in atezolizumab arm. And the last figure on the right show that higher is the degree of interaction for these cell phenotypes, and larger is the increase of PCR from the addition of atezolizumab. Notably, biomarker assessed by IMC outperform 61 gene expression-based immune-related signatures. Indeed, none of these signatures demonstrate a significant test of interaction. In addition, we found that these tests of interaction retain statistical significance after adjustment for PD-LIGA1 and stromathias. In conclusion, we demonstrate IMC is feasible in a large randomized trial and provides a comprehensive overview of triple-negative breast cancer heterogeneity at a single-cell level with spatial resolution. Bulk protein or immune-related gene expression signature do not consider the cell compartment of expression, and this may explain why they provide more limited predictive information. Consistently, we found, for instance, that assessment of Ki67 TME and HLA-DR in epithelial cells seem to offer more predictive information. We highlight the potential predictive role of PD-LIGA1 positive, hydropositive antigen-presenting cells, and CD56 positive neuroendocrine epithelial cells. High degree of spatial connectivity between epithelial cells and specific TME cell phenotype has predictive or higher PCR rate with the addition of atezolizumab, independently by PD-LIGA1 and stromathias. This means that spatial information matters for predicting immune checkpoint inhibitor benefit in triple-negative breast cancer. This spatial TME interaction outperforms cell phenotype density in predicting differential response to immunotherapy. This finding deserves confirmation in other clinical trials and with other chemotherapy backbones. I would like to thank patients and their families, the generosity of the funding agency, all co-investigators, but in particular the leading contribution of Raza Ali and the terrific work done from his lab group, in particular CC1. Thank you very much for your attention.
Just to remind everybody, all questions have to be actually posted. And while we do that, since I don't seem to have a question, I will begin with one. Did you see, obviously this is fantastic work, Dr. Giampaolo. It's fantastic, and I think it makes a lot of sense. Do you see in the different subtypes of breast cancer a difference in immune profiling in the TME? For example, would mesenchymal subtypes have more immune deserts? Do you see a difference according to subtypes?
Absolutely. So the tumor cell and the TME microenvironment are in a co-evolution and in equilibrium state. So it's not surprising that there is a strong heterogeneity of the TME according to the specific TNBC type. For instance, the LARS and the mesenchymal subtype, as expected, are much less infiltrated, and more frequently, that is what we have already presented last ESMO, is more frequently associated with a condition in which you find really few stromathias. But this analysis allows us to illustrate even further the heterogeneity within the quality and activation state of these T cells, even in the condition where there is inflammation, like for instance, in the mesenchymal stem-like. We found CD8 and CD4, but not some other specific immune cell like NK, plasma cell, and PD-LIGA1 activated antigen-presenting cells. So the level of heterogeneity within these triple-negative breast cancer cell type is even larger than probably we expect.
Excellent. As I don't have any questions, I'm going to continue talking since we have a few more minutes. Obviously, IMC is highly expensive. It's probably not, it's not scalable. Any ideas how you could translate this into something that's scalable?
This is a very good question because as a clinician, we aim to transfer this information to something that is potentially clinically useful. And the challenge is how we do so. On one way, we have to expect that technology will evolve. I would say 20 years ago, we consider microarray very, very expensive. It's a challenge for really doing this in the clinical practice. Now we don't talking even anymore about this aspect. So one, I would say technology will evolve and will allow us to do something that now we don't see as possible in the clinical practice. The second is to move this technology to the clinical practice also require a lot of work in the computational approach, because I would say even the presentation I know was complicated. The computational approach behind is even more complicated. So I don't want that the impression is that this is something simple. This is still something that can be done by really few high experience group. I would say that we see the potential of this approach when we have adjusted for the suitability to transfer this huge amount of information in the clinical setting, but there is a lot of work to do.
It's fantastic. You know, I do totally believe that single cell is the way to go. I mean, that's the cell-cell interactions, and there are different subtypes, and I was looking forward to your talk. Thank you very much. I have no questions.
I think we're running out of time.